Welcome back, everybody. Today we're going to be talking about the regulation of the cell division and cancer biology. So looking at how regulation of cell division is directly related to development of cancer and what actually causes cancer. When you talk about the cell cycle, students sometimes have this misconception that the cell basically can control or basically just goes through the cell cycle without any regulation or any control system. But there is a series of proteins and enzymes that directly impact the cell's ability to actually move from G1 to S to G2 to the M phase and restart the cycle. So the cell alone does not just randomly move through the entire cell cycle. It is a highly regulated process with a bunch of checkpoints and controls within it that's going to regulate the movement of those cells through the entire actual cell cycle. There are three main checkpoints within the cell cycle. So when I say a checkpoint, I'm talking about a point in time where the cell can check itself before it moves on to the next cycle. And if there's issues at that point, the cell can either try to fix the damage or basically end its own life and prevent that cell from damaging the rest of the actual body. So the three places that the cell can check itself is there's a checkpoint within the G1 phase. So during this G1 phase, the cell is going to make sure that it's grown large enough to actually undergo cell division. During the G2 phase, there's a checkpoint where it's actually going to be able to check to make sure that the DNA was synthesized correctly during the S phase. So there's a process to make sure that the DNA is duplicated correctly to make sure there's no missing pieces or any missing damage or damaged parts. And finally, there's a checkpoint within metaphase, specifically in the M phase, where the cell can make sure and check that the chromosomes are properly aligned single file in the dead center of the cell, in the equator of the cell. So those are the three main spots where the cell has the ability to make sure that everything is okay and that it can move on to the next part of the cell cycle. And if something's not okay, the cell has the ability to try to fix it. And if it can't fix it, then it's going to undergo apoptosis that we'll discuss in a second. With regulating the cell cycle, there's both external and internal regulatory proteins and factors. So externally, cells need to be able to receive signals to know when to divide. A great example is wound healing and bone growth. That there must be, if you cut yourself, there must be signals sent from the surrounding cells to those cells to say, oh my goodness, we have a damage. You need to start making more cells so we can fill out that cut. So there's external cell signals that can be sent to cells that say, you need to start dividing and actually make more cells. Another impact is contact-dependent inhibition. And it's really interesting. We really don't know all of the factors involved in this, but when cells interact with other cells and touch each other, they will no longer divide. So as you can see in the picture on the right, if you notice that all the cells form a single surface, a single line, one layer of cells. Cells do not like to grow on top of each other. So when they touch each other, they will not be able to divide anymore, and they'll create that single lawn, that single layer of cells. And you can see if they remove some cells, such if you cut yourself, the cells can replicate, make more cells, and heal over that empty gap. But cells will not normally pile up on each other and divide up on each other. There's also internal regulatory factors inside of individual cells. So there are proteins that, such as the chromosome replication checkpoint, where there's proteins that are floating in the individual cell that can control whether that DNA was replicated correctly. Can we fix it? If we can't fix it, what are we going to do? So there's both external and internal controls for the cell cycle. What happens to a cell that's damaged or made incorrectly? Thankfully, we have a mechanism called apoptosis, which is known as programmed cell death, or in other words, cell suicide. That if a cell has recognized that it's damaged or was made incorrectly, and there's no way to fix that damage, the cell can actually kill itself to prevent further harm to the organism, or it's also a very natural process during human development. So if you look at human development, and specifically our fingers and toes, we have these cells that are in between all our fingers and toes, almost like webbed feet. Well, over time, through apoptosis, those cells will naturally kill themselves and die, and will get rid of that webbing in between our fingers and toes. So apoptosis, this programmed cell death, is also involved in preventing cancers. So if the DNA or the cell is damaged, it can prevent it from growing out of control. But it's also a very natural process in just actual human development and creating the actual organism. So we can see two examples of apoptosis. We have the development of a tadpole into the frog, and obviously quite a few cells have died because there's a tail here, but there's no tail in the actual frog, so all of the cells in the tail had to die. And you can see here in this foot at the bottom right that clearly apoptosis did not work correctly because all of the cells in between these two toes did not die like they were supposed to. What happens when control is actually lost? As we discussed, that's called cancer. So cancer is when these body cells lose the ability to control cell division. So they're no longer able to regulate their own division anymore. So it could be that they do not respond to signals anymore. So the body is sending them signals, or other cells are sending them signals to either not divide anymore, stop dividing, I'm not telling you to divide, and they just ignore them and just keep on dividing. Or internally, those checkpoint proteins, those proteins that are checking inside of the actual individual cell, might actually have a mutation, no longer function or form properly. These ones can have a genetic link. So it is possible to have some genetic mutations that are passed along to you that increase your risks of developing cancer within your lifetime. And you can see here that the normal cell cycle just goes crazy, and the cells start piling up on each other and start dividing and doing what they're not supposed to normally do. And this improper cell division can obviously just keep on growing, and these cells are not usually properly made, so they don't have the same normal characteristics of a normal body cell, and that's one of the issues why they cause so much damage, so many issues to the human body. There are two main growth factors that control the cell cycle internally. So we're talking about internal regulation at this point. The two types of genes I expect you to know are proto-oncogenes. So these are normal genes that can become oncogenes. Onco means cancer, oncologists, that can become cancer-causing genes when they become mutated. So these proto-oncogenes have a role in stimulating cell growth. So when they're turned on, they can cause cancer. So too much of a good thing's a bad thing. So if these oncogenes, these proto-oncogenes become oncogenes because they're mutated, their DNA is changed, so they're always functional, always turned on, those are going to cause cancer because they're always making the cell divide when it's not supposed to. An example of that is known as RAS, R-A-S. The RAS protein is a protein that when turned on causes cell division. Conversely, you also have genes that are there to stop cell division. So we call these tumor suppressor genes or tumor suppressor proteins. So these are the proteins that are going to inhibit cell division. So that when they're turned on, they're stopping the cell from dividing. So the issue with that, if there's a mutation in them and they no longer function properly, then there's no longer any brake on the system and the cell is going to divide like crazy. So an example is P53 that we'll talk about in a second. It's been pretty much found that almost all cancers have a mutation in P53, this tumor suppressor gene that makes it non-functional, and that's why the cancer can grow like crazy. We basically already said this, that cancer is this unregulated cell division, so it's unrestrained, uncontrolled growth. So when you lose those checkpoints, that G1 checkpoint, that G2 checkpoint, that metaphase checkpoint, those are all basically gone, and there's no longer any brake pedal on the cell division. So the cells divide, and become a tumor and become a cancer. And we mentioned this P53 gene that has an important role in the G1 checkpoint specifically. So the P53 gene is actually going to detect DNA damage. And I'll go through an example of this in a second. So it actually has the ability to check for DNA damage and stimulate enzymes that are going to try to repair it. And if it can't repair it, it's going to then lead to apoptosis of that cell. And it's going to basically say, all right, we can't fix the damage, so this cell needs to die before it causes more issue. And as I said, all cancers have this P53 protein shut down. So they no longer have this P53 protein, and the cell just divides uncontrollably. Here's the example of P53 that I do expect you to understand some basics of. So you can see here, the top is the normal the top is the normal P53. So you have some DNA damage, so either some heat, some UV radiation, or some chemicals, some carcinogens, cancer-causing agents, are damaged your DNA, which is a very normal process. Thankfully, we have this P53 protein that senses this damage and can recruit repair enzymes, recruit things that are going to fix this DNA. If it can fix the DNA, then everything's back to normal. The cell can divide normally. The P53 gene and protein has done its job. If it's unable to fix that damage, it's then going to send signals and say, we can't fix this. There's no hope. This cell needs to go. So it can cause apoptosis to occur and going to destroy and get rid of those cells that are damaged beyond repair. Now, if you're a cancer that has an abnormal p53, then there's a mutation, that p53 protein, so it no longer does its job anymore. So that DNA damaged, there's nothing in the cell that can sense it anymore. So there's nothing to stop that cell from, that damaged cell, from just dividing and dividing and dividing. And while most cells might die in this process because there's something wrong with their DNA, some of these cells will just keep on dividing and eventually end up as that cancer cell, because that p53 protein is no longer there to try to fix the damage, and no longer there to stop the cell from dividing, and it's just going to divide and eventually become a cancer. One thing I do want you to understand is that there's many steps involved in developing cancer. So it's not just like one thing and you get cancer. There's many things that have to go wrong in that cell for it to become a cancer cell. So one of the things that has to happen is, once again, these growth promoter genes have to be turned on. So these oncogenes must be turned on to get that unlimited growth. Those tumor suppressor genes have to be turned off, so there's nothing to stop that growth. And you need to turn off that apoptosis process, that cell program death process, that cell suicide process, so those cells don't kill themselves before they can divide. And cancer cells are immortal, so they can divide forever and ever and ever. And interestingly, they also promote blood vessel growth, so they actually develop ways to increase the amount of blood they're getting to that area, so they get more nutrients, so they could just keep on dividing even more than normal. And there's two types of tumors that you might have heard about in your life. You have benign tumors, which do not spread to surrounding tissue. And then you have these malignant tumors, which are the ones that have the ability to spread and metastasize. So metastasize means that it can spread throughout your body. So benign tumors, sometimes they do surgery, sometimes they don't. It's the malignant tumors that they almost always have to try to fight, because these are the ones that can spread throughout your body and cause damage throughout your entire body. And that spreading process is called metastasis, or when a cell metastasizes. And some basic causes that you should already know: smoking and chewing tobacco obviously is not good. Radiation and sun exposure, too much radiation, solar, UV radiation can cause damage to your DNA and cause skin cancer, like on the right side. You have some defective inherited genes that we'll go to more in class. Some viruses can cause cancer that I'll discuss later in class. Obesity can cause cancer, and some hormones that can increase cell division can also cause cancer. So a quick basic summary: normal cell division, DNA is replicated properly, chemical signals start and stop the cycle, so it's regulated, and the cells can communicate with each other so they don't become overcrowded. Comparing that to cancer cells, you have mutations that occurred when the DNA was getting replicated. Those signals that start and stop the cycle are ignored, and the cells no longer can communicate with each other, so there's nothing to regulate that cell division anymore, and the cell just divides, and becomes a full tumor in the body.